Okay, I think we're ready to start. Welcome to this why and now seminar of functional MRI. I would just give the floor to our speakers, Jingyuan, Chen, and Saskia Bollmann. Thank you very much for being here.
Okay. Welcome everyone. Jingyuan, Chen, and I will introduce you to functional magnetic resonance imaging in the next hour. And we have a lot of material prepared for you, so we get the acknowledgements out of the way before they get forgotten, and some of the slides are based on contributions from these people, so thank you. So before we start about how to do functional MRI, I just want to quickly elaborate why we can actually do it, and for that we have to look a little bit into the functional brain architecture. So the human brain is a dynamic hierarchical system, and we have both present. So we have functional specialization or segregation, but also functional integration. And that basically means that when we measure our signals, that the location where we measure something can tell us something about the function. If you imagine that all your visual or all your neurons that process visual signals are distributed equally across the brain, they could still do the same, support the same function, but every time you do an fMRI experiment, just the whole brain would be on. But because our neurons are clustered in visual area, in the visual cortex, that tells us something about the brain function. And just to make that clear, so functional MRI allows us to locate signals or locate areas, which means that your research question should always be a question of, or should be answered by a coordinate system or a coordinate in the human brain. So the question of how something happens then needs to be found by an interpretation through these locations. So MRI not per se tells you, oh, how does the brain do something, it just tells you where it does something. And from that, you have to infer how things are happening. And importantly, we only see neural signals that change the activity over time. So not everything that is required to perform a task will necessarily light up. So if you think about a simple flickering checkerboard, then you see visual cortex because you modulate its activation, but you don't see all the higher areas because they're present throughout the whole task to maintain your attention, to keep you awake. So only signals or areas where you modulate the activity you can then pick up in an fMRI experiment. The other important thing is this is in correlation with a task, be it like an external task or intrinsic and resting state. So there's no definite causal inference for every signal you measure. So that is also something that has to come from your experimental design. So everything we measure is just a correlation in the first step, and then you have to make sure that it actually means something and it's not just coincidence. And just to give you a rough overview of some important functional networks, we have the basic sensory motor system, visual, auditory, sensory, and the motor system. And you can see they are very well localized. We know a lot about them. They're easy to study. They're also easy to study in other species. And then as we integrate this information and have this hierarchical setup, we go up the hierarchies. We started, for example, with primary visual cortex, and then we have visual association areas where the input then is interpreted, and perception becomes recognition. We perceive an object, a face, and that is through the hierarchy of the visual system, for example. Same is true with language areas, the motor areas, so everywhere this principle is repeated. And these very general high-level function then rests on very large brain areas that need to come together to for example, whatever, learning or memory, which then has this network and hierarchical aspect. Okay, so that is the preface. So we will introduce you to the contrast mechanism we utilize, how we acquire all signals. We will talk about signal noise characteristics, and then how do we have to analyze the data. And in the end, we will talk a little bit about new directions in fMRI. So the bold contrast, the bold is blood oxygen level dependent. That tells us that our MRI signal is dependent on the blood oxygenation level, which should already raise a red flag and tell you, oh, this is not a neural signal per se. So it's a very complex cascade until we we get a signal that that we measure with MRI. And the basic or the very short story is neural activity requires energy, and the brain consumes an over proportionally high amount of energy. So about 25% of glucose and 20% of the oxygen of the whole body, compared to its weight, which is only 2% of total body weight. So it's a very power hungry machine. But it doesn't have any storage capacity. So if you don't have a blood supply for 1 true minute, your brain's dead. And so it has this just-in-time delivery, which means that for every requirement, it has deliver glucose, oxygen, and all that stuff in time to the brain. And that allows us to measure brain function by measuring these vascular or physiological changes. And that's just to show you how the cerebral blood supply actually looks like. So in red, you see the arteries that supply the blood, and then in black, you see the vein that then drain the blood and the waste products and everything that's not interesting. As you will see later, we are actually measuring downstream effects in the veins and venules from neural activity. But the actually active, so this is other way around. So this is a comic of the capillary bed. So this is where the nutrients are exchanged. That's where it oxygen is released and CO2 is taken up, glucose is exchanged. So that's where the handover happens. And then you have arterioles and higher up the arteries. And on the other side, you have the venules and then the veins that drain waste products. And basically, following a neural stimulus or following a stimulus, the active part is here. So it's in the arteries and the penetrating arterioles, and that is an increase in blood volume. And here you can see following a strong stimulus, you see in the arteries pretty high change in blood volume. And you can also see that it's a rather slow signal. So we are talking on the time scales of seconds. It's not as fast as neuronal signals, which happen at the millisecond range. So that's the first thing to keep in mind. And then you have this passive response in the veins, where they just dilate because there's more blood coming in. It's not an active, or not as actively controlled. And why can we measure it with MRI? And the secret to that is the hemoglobin, which transports the oxygen. And basically, oxygenated hemoglobin is diamagnetic, and deoxygenated hemoglobin is paramagnetic. And following neural activity, we have this neurovascular response, which then increases our ratio of oxyhemoglobin to deoxyhemoglobin. So it means we actually have an oversupply of oxygenated blood. And that was found in that was the seminal paper where the bold effect was, that's what you have to cite, basically, when you talk about bold. And so the study from Ogawa and colleagues, and he basically showed that here you can see the intracortical veins. So because they carry deoxygenated hemoglobin, they have a shorter T2 stalk, and that gives you a contrast and a gradient echo image. And then he did not do any task or stimulus or whatever. He basically just gave them some gas, and which increased the blood flow. So that's a pure physiological response. But what you can see is that in the image, the veins are gone. And that's because blood flow was increased, which can lead to an increase in increase in oxyhemoglobin, decrease in deoxyhemoglobin, which leads to the loss in contrast or an increase in signal. And that's also another like important takeaway message. We measure a physiological response. It's a physiological signal, not per se a neural one. But yeah, so how do we measure it? This is just a 2-minute summary of a field in its own. But I've introduced the 3 most important parameters you need to know about when you set up your protocol or when you look at the protocol on a scanner. So to summarize, we want to measure local susceptibility changes, so we want to be T2 star weighted, and we want to measure fast. So I said we want to kind of like pick up something that happens on the range of seconds, which means that kind of like per slice as we go through the brain, we cannot spend more than a couple of tens of milliseconds there. And kind of like 13 years before that requirement was actually made, and people said, oh, we want to do fMRI, Peter Mansfield already had a solution for us. And he termed it echoplanar imaging. And here you can see kind of like the first gradient echo bold EPI images that studied a neuronal activity from 1992. And how does it work? So instead of kind of like standardly, you excite, you phase encode, you read out 1 line, you excite, you phase encode, you read 1 line. Peter Mansfield said, okay, we can just keep going. So you have 1 excitation, you start in the center of K-space, you go out to your K-max, and then you just keep encoding, keep switching the gradients until you've kind of covered the whole K-space in 1 go. And that gives you kind of like, that takes about 30, 40, 50 milliseconds, and that kind of like is the time it takes you to acquire 1 slice. And then you just repeat it throughout the brain, which means you can end up with a couple of seconds, you have 1 brain acquired. And basically, the contrast is controlled by the echo time you choose. So as you kind of like increase the echo time, you have more T2 star decay, which means you have more signal loss because stuff decays away, but you also have more T2 star rating, and that's what you're interested in. And the rule of thumb is basically to have your echo time set to approximately the T2 star of the tissue or of gray matter, which is a very rough number, so you have some leeway there. And the other thing I want to highlight is resolution. So most fMRI studies are unfortunately done at that level, but EPI images can look pretty nice. So basically, like we only like half the voxel size in each direction, but that means we actually have 8 times higher resolution in volume. And you can see that still, it's pretty tricky to like pick out the gray matter sharply. So you see, okay, this is all gray matter. You have like CSF here, but the kind of like cortical ribbon is actually still pretty small. So be aware that kind of like keeping things apart, like different gyri, is still a challenge. And the third parameter is obviously your repetition time. So in the fMRI analysis world, this is typically the time it takes to acquire 1 volume, whereas the for the physicist, repetition time is the time between a consecutive excitation of the same slice of spins. And usually that's kind of the same, but it can get muddled every now and then. And as I said, we kind of like looking at dynamic signals, and to characterize the temporal stability of our overall signal, 1 important measure is the temporal signal to noise ratio. And here's just an example here of our magnitude image. And then the temporal signal to noise ratio tells us like how noisy is our data stream, so our kind of like 40 time series. And on the one hand side, you don't want to have any noise in it, so you want like the TSNR to be high, but of course you're interested in dynamic changes. So if this is infinity, then your scanner is broken, or your brain. Yeah. Jingyuan will elaborate a little bit on this.
Okay, so Saskia has just introduced both contrast and acquisition. So we'll first see what fMRI images look like. So we're actually treating a 4-dimensional data, and this is from a very conventional acquisition. So it's in a low resolution and low TR, but it's basically what the images we will acquire. So with the technical advances nowadays, we have pushed the spatial resolution to sub-millimeter level, and also the temporal resolution to sub-second level. So this is really one major benefit of fMRI. It's really superior over other neuroimaging techniques in better spatial resolution. So it can help us target which brain regions are activated by specific tasks or involved in complex brain behavior. And the second benefit of fMRI is whole brain coverage. So this really gives us a system view into brain function. But fMRI also has several limitations. The first limitation is when there is a brief neuroactivation and there will be hemodynamic changes, and then we finally see a rise in fMRI signals. It will take about like 6 seconds to peak. So fMRI is really an indirect and sluggish measure of neuroactivity. And this can be reflected by the pulse spectra. If we just plot the pulse spectra of some fMRI time series, so you can see it's really in the low frequency band. Not just because they are all in low frequency bands, they also look slightly different. So if we plot the autocorrelation, you can see some like regions have more dispersive autocorrelation and some have a narrower dispersion. So this is because our vascular anatomy is quite heterogeneous. So even different brain regions have the similar neuroactivity, the profile of the fMRI signals were a bit different. So this is a second limitation of fMRI signal. It's really confounded by the heterogeneous vascular responses. So it's hard for us to interpret, and the differences we see in fMRI is due to neuronal differences or those vascular differences. And the third limitation is it's really a contrast dependent on the hemodynamic changes, basically the ratio between oxygenated and the deoxygenated hemoglobin. So there are lots of other physiological processes will also alter the ratio between these kinds of hemoglobin and lead to differences. So in that regard, fMRI is more modest in neuronal specificity compared to other imaging modalities. So after seeing the fMRI signal characteristics, we'll go over some basic noise types. So as it's similar to a previous talk by Jason Stockman, for fMRI signals, it will suffer from some common noise type like thermal noise and system noise for common MR images. So I will just briefly touch on the motion and the physiological noise. For motion is because we try to monitor the temporal dynamics of the hemodynamic changes of each voxel. So we really want the time course represents a value from a single location. But unfortunately, subjects tend to move within the scan, especially when the scan time is pretty long, or we are scanning pediatric patients, or it's kind of diseased population. So here, just showing a movie of a subject like inside scanner, you can see there are lots of motion there. To correct the motion, the first type of like artifacts is really to make sure all the voxels are aligned with each other. So it's really done through rigid body registration to a reference volume. So we make sure the time series of each voxel are correspondent. But this is usually not adequate. The reason is there's also intensity changes associated with motion. It's called like spin history effects. So basically, when we image fMRI data, we first excite things of one particular slice and then acquire the data. But when we excite a particular slice, the subject has moods, and when we acquire the data, the spins we're collecting are different from the spins we excite. In that case, there will be intensity change in the fMRI signals. To accommodate those changes, so sometimes people will include the motion parameters, basically 3 translational direction motion, and 3 rotational direction motions in your regressors and projected out of the data. But just remember, any retrospective motion correction method is never perfect. There are continuing interest in doing prospective approaches, basically monitor the motion movements in real time and incorporate into image reconstruction. It's still not very sufficient. So the most effective way is really to make sure or minimize head movements during acquisition. This can be done by proper padding or using customized head case. The first type of physio noise is actually also related to motion. So the figure here just shows the images progression across time. You can see even the boundary seems very stable across the entire scan. The tissue really moves across the whole time. This is related to the cardiac and respiratory cycle induced. So if your TR, the sampling rate is fast enough, you inspect those time series, you can see those sharp peaks from the respiratory cycles and the cardiac cycles. So regions are most affected by this kind of motion are more localized to large vessels. So image here kind of shows those regions whose percent signal variance is most explained by those kind of positivity related. I just want to highlight why this is important. For instance, for insula, I think lots of people are probably interested in some emotional cognitive functions, but then it's really those regions who kind of suffer most from the positivity-induced motion. So that's kind of some important thing to keep in mind. And so how do we generally reduce this kind of motion? If your TR is short enough, so you can resolve the peaks and you just do low pass filtering. But in cases your TR is pretty long and we're aliased into low frequency bands, one common approach is based on modeling from external physiological recording. A key assumption is if this acquisition point sits in a fixed phase during one cycle, so how it's going to be affected by these artifacts will be similar. So it just give you some idea how it's kind of implemented. I wouldn't go into too much details. So basically you have an external measure of your pulse. So you know when there's a peak in each cardiac cycle. So you treat each cycle as a 4 to pi phase, and then you have the acquisition images. So for each TR, you calculate which phase it is in the current cardiac cycle. So you get a time course of phase. And then you, based on the phase, you generate lots of sinusoids kind of representing the physiological fluctuation related to it. So, and then project out the data. That's just basically the idea. And then why that's important, because some like high frequency physio fluctuations are riding on top of your low frequency functional signals. So if you remove them, it will mitigate your noise levels. So it will enhance your statistical scores. So this is just an example from a sensory task. After you do this physio correction, you really see a great increase in all the signals. But if you monitor those physiological recordings, so this just from one subject, and the red curve shows the respiratory belt, you can notice its envelope. It's not very stable over time. It also have very slow fluctuations. And if it plots pulse spectra different from those kind of high frequency respiratory peaks, it resides in a very low frequency range. So these are actually not just noise. They kind of reflect some slow changes in the systemic physiology that's regulated by the autonomic nervous system. For instance, people have argued those changes in the amplitude of the respiration kind of related to the entitled CO2 level, and CO2 is a vasodilator. It will cause blood vessels to expand. That will kind of contribute to more blood flow. And similar for heart rate variability. And it's kind of important is because this signal property really very similar to fMRI signals we see. And then they're also in the very ultra slow regimes. And unfortunately, it affects most gray matter areas. So this is really a very scary figure. So for this subject, it has very gigantic motion changes in the envelope of the respiratory trace over time. And then you can see here, just plot voxel-wise fMRI time series. So each row is one brain voxel, and then here gray matter, here white matter. And you can see the amplitude changes really follow those respiratory measures. And then it will naturally confound your fMRI measurements. For instance, here you want to assess a functional connectivity. We will go into it later. Basically, you pick a C region and assess the temporal synchrony between the time series of other regions in this particular region, and affect the correlation or temporal synchrony values. This is a normal case, and all of a sudden you see, oh, whole global signals are coupled with each other, but it's not necessarily a big state change, and it's possible if there is just a deep breath. So how can we get rid or remove this signal? I just give some high level idea how we kind of people address this kind of physiological fluctuation. And the first idea is kind of model-based approaches. So you have external physiological recordings, and then you achieve those measures of those physio signals. And then you convolve it with some previously published physiological response functions characterized in early studies, and then linearly project these signals out of the data. Another type is really anatomy-based approaches. They're assuming most neuronal signal are in gray matter. In white matter is dominated by those physiology and then non-neural sources. So they kind of creating masks from those non-gray matter regions and then extracting signals from it. There are kind of more emerging and also more sophisticated ways based on multivariate data-driven decomposition. So basically you have 4-dimensional fMRI data, and based on some prior assumptions or hidden assumptions you made, it kind of simultaneously decompose it into multiple components, which have a spatial pattern and temporal pattern. And there are certain criteria for you to really choose which might be related to physiology. And then after identifying it, you kind of project out of the data. But physiology is really a very debated issue in fMRI. So by far, we are treating it as a nuisance factor, but we know it's regulated by the autonomic nervous system. So it could be actually reflect actual neuronal inflammation. So for instance, in this study, the subjects are doing a lexical decision task. But if you look at the activation pattern, lots of region lineup actually doesn't belong to the region you are think to be involved in decision making. And if you monitor the respiratory changes over time, and you can see it follows the task paradigm pretty well. So in those cases, sometimes subjects will be task coupled respiratory. It's really hard to tease neuronal activity and physiological activity apart. And here is another example is like during sleep. So these are just EEG signatures when those points here, some like EEG signatures during sleep. And then you can see it's kind of when it's occurred, there will be a drop in the amplitude of your pulse measures. In this case, it's also physiology and neural activity are coupled. It's hard to separate them apart. So you don't know the fMRI signal you see is really arising from neuronal mechanisms or those consequences of those physiological changes. I think in Martinos Center, we're doing pain study. This is from the lab. So people are really kind of torturing subjects using a finger crush. So to induce pain. And the top pain is ideal response derived by convolving the kind of paradigm with the canonical hemodynamic response function. And the bottom, the left panel shows the heart rate changes, and right shows the heart rate variability. And you can see in this case, there are really kind of task coupled changes in the heart rates. So if you don't think more about such a coupling, just go ahead with all those physical correction. Lots of activation actually gone after you do this correction. So it's really hard. I don't feel there is a kind of good solution to it currently when there is task-coupled physiological effects. But because it can have such a dramatic effects on your data, it's really important to kind of keep checking whether your physio measures are coupled with the task, especially in those cases you expect to happen, for instance, like stress, arousal, or like pain-related studies. Okay, so I would just by far mention there are so many like noise in fMRI data. How do we identify interesting information out of the data? So first is like we need to do preprocessing because fMRI data is just so noise, and then we're going to make lots of assumptions about the data when we do further statistical analysis. So the goal of preprocessing is really just to make sure the assumptions are met and also kind of improve the SMR. There are lots of common steps of preprocessing. I wouldn't go into too much details because I think there will be a later talk in the why and how focused on advanced analysis of fMRI signals. So here we just basically give overview. Apart from those motion correction and physiological noise correction, I just want to highlight which preprocessing step are important really depending on your applications. I'll just give two examples. In one case, you're interested to use fMRI to infer causality between different regions. Most of the causality analysis are depending on the temporal delays between two remote regions. But for fMRI, unfortunately, not every slice are required at the same time. So sometimes this is kind of conventional acquisition. So either going ascending order or interleaved order. And nowadays we're using simultaneous multi-slice. It's in a more complicated fashion. But if you, for instance, have a TR of 2 seconds and you have 30 slices, from the top and the bottom, there is a delay kind of up to 2 seconds. So it's going to confound your causality inference if you don't correct for it. And fortunately, if we know the slice timing, we can just like do a temporal interpolation to put it back. And another message we want to convey is like, your preprocessing can really have a huge effect on your data. So for instance, about spatial smoothing, the goal for spatial smoothing is to improve SMR. So the signal will be kept, but then the noise will cancel out. And sometimes to improve the registration across subjects. So the general idea is just to replace voxel with a weighted average of nearby voxels. And you can see when you increase the kernel of the like smoothing, and then you will get more blurred image. So here just give some indicative about how smoothing are going to affect your activation. And then with the change of smoothing kernels, you really see a quite big difference in your activation pattern. And what's the optimal smoothing kernel? There is a theory saying like matched band filtering. So basically, if you know the intrinsic activation resolution of the data and just apply the same resolution, but it's really hard to know which is intrinsic resolution. So it's another question. There is no optimal solution to it, but you kind of being aware or you have some idea about like possible resolution in your data. And like to final remarks about preprocessing, it's like not everything is always done. For instance, if you really care about or high resolution data, you spend so much effort acquiring those high resolution data, you actually don't want to smooth it to drop out your resolution. And also it can be done in other orders. It really depends on your study goals. So to go with analysis, we just go back to what's the benefits of fMRI. So compared to other imaging modalities, really it has better spatial resolution. So that means it can localize which brain regions are activated. And also it has whole brain coverage to look at large scale like organization or interaction between different regions. So there are lots of types of fMRI analysis. I just briefly covered 2 types of it. And the first is what I just mentioned, using the role or leverage the high spatial resolution to see which regions are activated by specific sensory or cognitive behavior. And second is to use fMRI to infer local and distributed functional connectivity. So for the first type of analysis, this is still single voxel based. In that regard, each voxel kind of react to the task separately, but it's not likely the case. So there is kind of emerging interest in doing multivariate analysis, just basically show what's the pattern of how different regions kind of react to the task. And also for the second, we look at like functional connectivity, temporal synchrony between different regions. They're still assuming these regions are spatial over time, but there may be different spatial temporal patterns. So there is a huge interest or a trend kind of going to explore the spatial temporal dynamics associated with the multivariate analysis. But today I'll just briefly talk about the first 2 types. So to identify which regions are activated by task, so for fMRI really just depend on the blood oxygen level dependent contrast between conditions. So let's see which regions are activated or are different, show different behavior between these 2 conditions. We just measure fMRI signals across entire scans, and we sort all the signals from one state and sort all the signals from another state, and then we compare the signal intensity voxel-wise. And then we get a statistical map, and then regions demonstrating significance, we consider they to be related to the task. And that's what you commonly see in fMRI kind of publications. It's kind of those rare regions highlighting those task-related regions and overlay to anatomical image. So this is just an oversimplified representation of the concept, how we imagine which regions are activated by the task. And I think probably in the later, more advanced fMRI statistics talks, you're going to hear how this idea will be incorporated into a general linear model. So just highlight some applications. For instance, these 2 map very detailed like visual fields, and not just like in sensory regions. And in this case, they target which regions are involved in the working memory function, and also like show how depressed subjects have a typical activation in the reward system, just as potential for biomarkers. Okay. A second type of analysis is basically based on the system level view of fMRI to characterize local and distributed brain functional connectivity. So here it just portrays the signal intensities during a resting state, basically no task state over time. So the red color means positive signal fluctuations and blue color means negative signal fluctuations. And then they're just not random noise. So if you compare the time series from two remote regions, you can see there's some synchrony there. And then it's always quantified or commonly quantified by linear Pearson correlation. So the higher correlation, they assume these two regions have more connectivity and likely to be involved in one functional network. So that's kind of another dominating or widely used analysis nowadays. So by using this approach, people have identified during the resting state, there are multiple networks operating simultaneously during in the brain, and different colors just have those broad regions kind of involved in one network. Basically, mathematically, just these regions have a very high temporal with each other. And then not just during resting state, and people start to quantify those temporal synchrony during more cognitive task conditions. And then nowadays, people are interested in like naturalistic stimuli and how this connectivity will be altered. And then also how this can serve as clinical biomarkers. In this study, they show in Alzheimer's disease, so there's kind of reduced activity in the medial frontal cortex. And there's also interest in how we can use fMRI to characterize connectome of each individual subject and distinguish the behavior. Okay, so I think we currently have like 42 minutes. We probably will have another 10 minutes to highlight some like new fMRI. But I think Saskia and I would like to take some questions if you have any. Or we can just continue.
Everything clear.
Okay.
Ah, that one. Sorry?
Are there any other types of basic?
Oh, yeah.
For instance, like a
I think EPI is just the most easiest to use. So what's like from a gradient side, performance side of view, from a reconstruction side of view, it's very close to everything else. I mean, there are other trajectories like stack of spirals, styles, whatever. But the effort, at least until today, to kind of get a decent image out of it is kind of too high to then do all the other stuff that is also involved with the fMRI study.
I think both spiral and like EPI, those are pretty common. They have different artifacts. It's just that nowadays EPI becomes dominating. It's very easy to implement the simultaneous multi-slice. It's a very challenging for spiral to do that.
Yeah.
How many series?
I don't know, I mean, you can do the math. I would say like half a second TR, and the typical task goes somewhere between 5 to 10 minutes, or kind of like one run. So you have, whatever, 600 up to 1,200 images, and then usually kind of like people repeat these runs. So you generate a lot of data very easily.
So slightly slower.
Yeah. But you also scan a very long time, so. Okay. So, yeah, kind of like the parameters I mentioned, kind of like half a second TR. So, I mean, EPI and MRI are pretty young techniques, I would say. As I said, kind of like the first images were 30 years ago. Yeah, 30 years ago, 28 years ago. So the hardware and the reconstruction and everything is kind of like still evolving, and we basically moved from maybe a TR 6 seconds and 4 millimeter resolution, probably something like half a second, and 1 millimeter resolution. So things are still constantly developing through changes in hardware, reconstruction, and everything, which kind of like also allows us to explore the signal we measure from a different view. And like with the introduction of simultaneous multi-slice, people came to realize that when you measure a signal at higher temporal resolution, you also obviously see higher frequencies, and they found neural activity also at higher frequencies, so above the 0 Hz that were kind of like traditionally assumed to be the cutoff for the bold response. I mean, we know that neural activity is faster, but people always thought, okay, the bold response is really slow, so we can't see anything. But Laura Lewis, who is now at BU, she really showed that even for kind of like stimuli with 0.75 Hz, you actually kind of like get a bold response associated with it. And people are also in the resting state literature looking at that. So like similar to EEG, you split it in different bands, and you still see kind of like significant correlations across brain regions. And yeah, but that's kind of like pretty new development actually means. One of the kind of like nice advantages is that we have actually a cleaner representation of our signal. So traditionally, you kind of like probably had the cutoff somewhere here, and all the kind of like noise, like respiration and cardiac activity, was then kind of like meshed into your neuronal activity as well, and it's kind of like pretty mixed up. But kind of like increased bandwidth, we can better sort out these components we are not interested in. And you also kind of like get a boost in statistical power. It's not like a one-to-one thing, but kind of like going from a TR 2 second to 0.8 second, you can see how kind of like you're more sensitive to activity, and then going further doesn't really change anything. And that's because bold signal is still correlated in time. It still has an intrinsic lower frequency. So adding more samples doesn't necessarily add more information into your data. So there's a sweet spot to find on where you will gain and where you just make your life harder. And kind of like going in the other direction is kind of like increasing spatial resolution, which was especially driven through the development of 7T. You see things like 3D EPIs, where you can really acquire not whole brain coverage, but a slab across an area in like 2 seconds, maybe 1.5 with sub-millimeter resolution. So kind of like really dialing down these fine-grained structures. And the obvious kind of like advantages, a reduction in partial volume effects. So if you, for example, look here, then you see kind of like how the banks of the circles are just squashed against each other. And in EPI space, they're basically in one voxel. But of course, because we know that they belong kind of like to different areas of the brain, functionally, they're very, very different. So kind of like resolving these differences becomes where the high resolution goes. And we also slowly approach kind of like the fundamental functional units of the brain, like columns and laminae. But you can see that then the analysis of the data has to change. So it's not your standard GLM univariate test. You really have to kind of like think about what do I measure, how do I kind of like aggregate the data so I get my SNR back. And just some math. So kind of like the human cortex is about 2 to 4 minutes. And from histology, we know that we kind of like different cells and the 6 layers. So you know where we need to go at some point in time. So currently we have maybe 2 to 3 voxels within kind of like the cortical sheet. Similarly, cortical columns. So we're not there quite yet, but we're getting closer every year. And this one stripes into, that's not a cortical common part now, but it is approaching that level of resolution. And here on the right, you can see kind of like individual fingertips resolved in sensor motor cortex. So that's something we can now start to look at. I'm handing over.
So the second direction try to touch is about multimodal fusion. We view it as two primary directions, and the one is a combination with neuromodulation techniques. So there are lots of animal or like human kind of neuromodulatory tools really can excite or suppress the function of specific brain regions. So when it comes to fMRI, will allow us to probe the directed interaction this region might have with the rest. So just give one example. This is a TMS-MRI. For instance, they want to see if the executive control network and salience network have some hormone network. So what they do, they use TMS to excite these two networks separately, and then assess between default hormone network and this network, and to show if there is some salient difference. So if they can view some difference, as shown here, really indicate there is a causal relationship between the excited network and then the targeted network. Another direction is to integrating fMRI with complementary modality. The idea is that fMRI has high spatial resolution, but it's not direct measure of neuron activity. Lots of other more direct measure of neuron activity, but they don't have whole brain coverage and high spatial resolution. For instance, scalp EEG and the calcium imaging rodent studies and electrophysiology. And for that, even if you have whole brain coverage and accessibility to those neurochemical conditions, or spatial resolution than MRI. So here just one example of how when random you can retrieve lots of information. So EEG signal can be used to delineate brain states. So here is wake and deep sleep states. So then you characterize function for these two states. There's another recent science paper the global dynamics present different patterns. Another benefit of integrating these complementary modalities really also can improve our interpretation of fMRI data, because we have other measures being more direct measure of neuroactivity. I will give two examples. So this is really a seminal study from Lockfed in which they demonstrate the neurophysiological correlates fMRI signal. So they measure several recording, including local field potential, multi-unit activity, and single-unit activity, and compared to both fluctuation, which is the shaded area shown here, and then potential have prolonged response, kind of matching the timing of both. So they kind of position that compared to other modalities, other signatures, local field potential kind of more related to both signal. And another example is how we use EEG to help interpret the functional connectivity. In this case, you have EEG signature, and you can based on to identify when the subject's alert and when the subject's more drowsy, and you compare the connectivity pattern. For instance, you can see when people are drowsy, there are more global correlations across different cortical regions, and then when people are alert, the function of different networks seems to be more segregated in this case. And the final slide, we just briefly touch it because fMRI is one of the most available neuroimaging techniques. So there are lots of data initiatives. So for instance, Martinos Center is involved in the Human Connectome project. And for the Bollmann project, it's kind of going to recruit 5 million people. It's a really very big data set. So there are lots of kind of exciting opportunities because big data, we are going to kind of have higher statistical power. And also a major appealing part of this project is like for such a large population, people know there are certainly certain percentage of people going to develop different kind of disease in the later phase of their life. So if we keep imaging them, so we will have the precious data keeping tracking of their imaging data when they progress to different disease. But also pose lots of challenges. It's like we have so much rich information. How are we going to release the computational load storage, and how are we going to mine all those complicated information from all this data? So these are very active areas in fMRI nowadays. So with all those introductions, and we finally just have a slide about the analytical toolboxes. There are so many analytical toolboxes available, and we just highlight a few if you are interested to explore. And then there are very good tutorials, like give very comprehensive view about functional MR imaging. We just list 2 of them. With all that, just thanks for your attention, and we're happy to answer any questions.